thanks a lot, and thanks for the invitation. It's really a pleasure to be here. So I actually have a slide about this. I'm originally from Hungary, and I studied computer engineering there. And I started working on computational systems biology during my PhD, which I did in Singapore. And about 4 years ago, I came here to Boston and started working in the laboratory of systems pharmacology, which was set up around that time by Peter Sorger. It's at the Harvard Medical School on Longwood Avenue, very close to here. And the aim of the LSP is really to try to come up with new conceptual ways of drug development by understanding cellular mechanisms in a systemic way to improve drug design. And the work that I'm going to talk about today is actually funded by DARPA. The first big project that we worked on is called the DARPA Big Mechanism Program. And since then, we've written and won a number of other follow-up grants that branch out from that basic idea, also funded by DARPA. And so Big Mechanism started with this idea of trying to be a complement to big data in that it tries to pull together automatically knowledge and mechanisms from various sources and then assemble them into predictive and explanatory models, which is, you will understand how it's different from big data since knowledge is collected and assembled rather than derived de novo from data. So that's where this work started from. Now our lab focuses on therapeutics, and in particular cancer treatment is one of the main case studies that we're working on. This picture is from NCI, the National Cancer Institute, and it tries to show the current paradigm of precision medicine. So you see how you have different populations of patients, each with a specific trait, which is typically understood as a set of genetic mutations. And in an ideal case, there's a specific drug that targets that trait, again, usually a specific genetic mutation, and kills your cancer cells and lets you survive. And the molecular biology, very simplified molecular biology view of this paradigm is that here you have a cell, this is the cell membrane, and there's a linear path from which signals are transduced in the cell and lead to cell growth and other phenotypes that cause cancer. And if you have a targeted drug to block this path, then you can stop cells from growing. Now the problem is that the reality is more similar to this, where you have a much more complicated set of microenvironmental factors that the cell is subject to. You have a huge network of highly interconnected pathways that have as many crosstalks as intra-pathway connections. Drugs aren't perfect and they hit multiple targets with different strengths. Cancer patients typically have hundreds of mutations, not just one mutation, and it's very often not clear which single one drives the cancer. It's probably a multitude of things that together constitute to cancer growth. The cell adapts in different ways to environmental stimuli and drugs. It can change its gene expression and change its behavior and compensate for various effects and secrete molecules that then affect cells in its environment to behave differently. So, and in fact, my argument is that to a large extent, the reason why the development of novel drugs, and in particular the development of novel cancer drugs, is so inefficient is because the basic understanding of these molecular mechanisms is not there to a sufficient extent. So we use instead these simpler rules of thumb to target cancer rather than deal with this.
Is that due to lack of data or to lack of understanding of such complicated structures?
To some extent a lack of data, but also an extreme fragmentation of knowledge in molecular biology. I'm going to come to that later in my talk, that there's about 1.2 million new publications every year in biomedicine, and simply collecting and keeping track of that information and assembling it into something that lets you reason over that information is extremely difficult. And so there's a lot of untapped knowledge out there that hasn't been collected and assembled systematically. Data is another factor, yes, but there's, yeah. And you probably all know this story that the majority of drugs actually fail sometimes in the very expensive later stages of clinical trials simply because they don't work as expected. All right, so systems biology is a subfield of biology that tries to understand cellular behavior as a system, so building up from elements and interactions into pathways, and then a systemic understanding of cellular behavior. And several modeling methods have been developed to pursue this kind of approach to biology, and I'm going to introduce one specific one, which is, assume you have a small molecular system like this, a receptor that phosphorylates a given protein, which then dimerizes, translocates into the nucleus, where it changes the cellular behavior, the gene expression, and then it dissociates, translocates out of the nucleus, and is dephosphorylated and completes this cycle. You can model a system like this using a set of differential equations that are derived from the chemical reactions underlying these molecular changes. The things that are in square brackets are concentrations of chemical species, and these K constants are actually rate constants of chemical reactions. And if you have a model like this, then given some stimulus, which is this time course profile of Apple, you can actually simulate this model, solve the differential equations, and get a time course prediction for the behavior of all of your biochemical species. So this is one type of systems biology model that people use, and there are many other. But at least this gives you a feeling for the kinds of things that I'm going to talk about. And this example from the previous slide is actually an example of bottom-up modeling, where you have some understanding of detailed mechanisms, and starting from these detailed mechanisms, you build up a model of multiple, by putting together multiple mechanisms to explain a specific phenomenon. And the issue here is that this type of modeling doesn't really scale very well, because these models are traditionally manually built, and it can take on the order of months or sometimes even years to curate one of these models. But on the other hand, a lot of the data that is currently generated is very high throughput. You have data for dozens of conditions and potentially tens of thousands of readouts, like the phosphorylation state of every protein in a cell. And that type of data is rarely analyzed with this kind of methodology because it doesn't scale, and rather people tend to use sort of model-free, clustering, various kinds of machine learning approaches that don't really make very good use of the huge amount of mechanistic knowledge that is actually there about these molecular species. And so the work that I'm going to talk about tries to really bridge some of this gap by trying to automatically assemble models like this at scale by collecting and assembling information automatically, so that one can then apply it to a data set like this. So one way to do that is to try to make use of the literature out there. You can see it here that PubMed roughly has 20 million publications from various fields of biomedicine, and a lot of these are talking about detailed molecular mechanisms like phosphorylation potentiates alcohol-mediated ternary complex formation and transactivation. So this is like one mechanism described in great detail that you can, if you could extract and assemble it into your models, then it would contribute to your understanding of molecular biology. And there's about 1.2 million of these papers per year, and about 20% of those are really at the molecular level, and so one way or another are relevant for cancer biology. So that's hundreds of papers per day that come out with new knowledge and discoveries in this field. So the system that we built, here's a conceptual diagram of how it works. We're trying to draw on various kinds of input knowledge, pathway databases that are manually curated by experts, literature, and also expert input, which I'll talk about a little bit later. Then there's a layer of knowledge assembly where all these pieces of information are collected and assembled in a way that one can then turn them into various kinds of models. So executable models which lead to simulations, or mechanistic networks which can be used for structural analysis or causal pathfinding, and so on and so forth. And data can come in various stages of this assembly pipeline. One way to take data in a specific context, like a specific cancer type, a specific patient, into account is during the course of assembly. But one can also use it to calibrate models. So you take an executable model and you calibrate its parameters to meet the observations in this given context. And putting this all together, you can produce explanations and predictions in a model-driven way to answer a specific set of queries. To elaborate a little bit on this knowledge representation and assembly idea, so in this assembly layer, one needs to represent information from disparate sources in a standardized form. One needs to be able to correct certain types of errors that creep in when processing these sources. One needs to be able to find and resolve complex redundancies across sources, various different ways in which people describe not exactly the same but related concepts in the literature, for instance. One needs to be able to assess relevance to a given problem, assess believability, that is what is likely to be just a machine reading error or that there's no sufficient evidence for altogether to be included in the model, and in some ways infer missing information that isn't explicitly stated in these sources but can be inferred from putting pieces of information together. So we've implemented this idea in a piece of software called INDRA, the Integrated Network and Dynamical Reasoning Assembler. There's this landing page at indra.bio, which also links to the GitHub page where this is available. It's a Python package, you can clone it or install it, and you see how the software architecture really matches the conceptual framework. So on the left-hand side, you have these APIs and input processors to various machine reading systems and also pathway databases. I only show 5 here, but we actually have 15 more of these, so we have a lot of these input sources. In the middle, you have INDRA statements, which are the internal knowledge representation of INDRA. And these red circles, they are all internal assembly components, which are, again, doing these kinds of tasks. And on the right-hand side, you have the various model output assemblers. And ISD, I'm not going to have enough time to talk about that, but that's basically the framework that allows us to assemble these mechanistic dynamical models. But we have a number of assemblers in various network formats. CX, for instance, is becoming the standard for cytoscape. SBGN is systems biology graphical notation, so this can be used for various kinds of network displays and network analysis. And we even have an assembler for English language, so you can take some knowledge and reassemble it into English and create little snippets of text or even some things that look like publications from this internal machine representation of knowledge. Okay, so I'm going to first talk a little bit about the various knowledge sources that we use. So one big source of information is pathway databases. Various funding agencies have been funding efforts to try to manually curate knowledge from publications and data sets into machine-readable structured sources. Pathway Commons is probably the biggest aggregation database that uses a format called BioPax to standardize information across multiple sources. The creator of Pathway Commons estimates that only 1 to 3% of published knowledge is actually in this aggregated database. And it's subject to several fairly important issues, these sources aren't actually assembled in the sense that their cross relations are represented. They are just dumped into this common representation format, but the relationships between potentially redundant knowledge aren't really resolved. And bypass edges, I'm not going to get into that, but it's the distinction between direct mechanisms and indirect effects that aren't always differentiated by these sources, and that can cause a problem in modeling. There are human-curated sources that use a very different kind of idea for curating knowledge. One of my favorite ones is BEL, the Biology Expression Language, which was developed by a Boston area company called Celventa. And they have a publicly accessible corpus of about 80,000 manually curated statements. And it's very interesting because it combines molecular level information like this one, which talks about a protein phosphorylating another protein, and higher level processes like the biological process of exercise, like actually doing exercises in the gym, increases the kinase activity of the MAPK1 protein in skeletal muscle, right? So it is able to tie together very different levels of concepts and represent observations in this way. Now, in terms of the machine reading systems that one inputs natural language into or goes out and reads full publications with, one of the main systems that we are using was developed at the Institute of Human and Machine Cognition in Florida. And it's a deep semantic reader that is actually a general English language reader. It is just loaded with dictionaries and ontologies relevant for biology so that it can recognize the named entities like ASPP2 and METK1. So you give it a sentence like this: ASPP2 can be phosphorylated at serine 827 by METK1, and it gives you a semantic meaning representation graph, sometimes also called the logical form. And you can then traverse this graph and take out the arguments that you need for representing in INDRA. So you see things like there's a phosphorylation event, and you can see the location of the phosphorylation, you can see the agent and the affected arguments of phosphorylation, and some other meta information like it's a passive sentence in the present tense and so on and so forth. A completely different machine reading system was developed at the University of Arizona by Mihai Surdeanu's group. Some of you might know the Stanford Core NLP system. Mihai is one of the developers of that system. And this system is actually using something that is like a regular expressions plus. So the system does a simple syntactic parse of a sentence, and then it defines these human curated rules over those syntactic graphs to extract events. It's much more shallow than this previous deep semantic parse, but it's a lot quicker. And assuming that one can cover enough ground by manually curating these extraction rules, it can work quite well. So these are just two examples, but INDRA is actually integrated with several other machine reading systems that all have completely different, stories and approaches to doing machine reading. There are some that are completely machine learning based, and so on. And more recently, we've actually been generalizing INDRA to not only biology but other types of causal mechanisms, and so it's also integrated with 4 general purpose causal relation reading systems. Okay, so now the internal representation of INDRA is a little bit.
Question. So are these reading full text or they're reading the abstracts at this stage?
Well, that's an orthogonal question, so you can put any text into this. We actually have access to about 35% of full text content through PubMed Central, which catalogs a lot of full text open access publications through licenses from Elsevier. But about 65–68% of content we only have available as abstracts, and then we could read them as abstracts. That's a very interesting subtopic of how to access this literature content. I can talk more about that. Okay, so this is a little bit in the weeds, but this is basically internal internal knowledge representation of INDRA, the UML diagram of INDRA statement. The main point here is that we can represent a large number of molecular mechanisms that are relevant for capturing knowledge about how cells function. And each of these statements has a number of agent arguments, which are like proteins or chemicals, things like that. And these agents have pretty rich states. So for instance, for a protein, we can represent its modifications, its mutations, whether it's bound to anything else, whether it's active, its cellular location. And as an argument of an INDRA statement, that would mean that this protein has to be in this given state to take part in that mechanism. Okay, now, given a set of INDRA statements that we collect from different sources, we have to do some knowledge assembly. And some of you might be familiar with Genome Assembly, where you have this device that sequences the genome, but it's only able to sequence little pieces of the genome. So you end up with these little snippets. And there you have to use some kind of an algorithm to align these, find redundancies, and align these pieces so that you end up with a single coherent gene sequence that you can use. And knowledge assembly is in many ways very similar to this, and here are some examples. These are written in English, but they map basically onto INDRA statements. So you have the protein MEK phosphorylating ER, 3 times from 3 sources, but then you have things like mistakes, reading mistakes, like accidentally a reader picked up ER phosphorylates MEK instead of MEK phosphorylates ER, or it didn't correctly resolve the named entity and it accidentally thinks that it's a chemical instead of a protein. And then you have versions of this knowledge that are at different resolutions. So MEC1 is a specific type of MEC, because MEC is actually a protein family. Sometimes they specify the specific amino acid site at which this phosphorylation happens, in other times they don't, and so on and so forth. And sometimes there are mistakes in that site. And at the end of the day, what you have to end up with is this piece of information that contains the most mechanistic detail possible and is non-redundant, and that's what you can put into your model. If you just take this output and try to build a model, it's going to be a completely incoherent model with all kinds of causal gaps and bypasses. So this is really what the internal assembly components of INDRA are trying to solve. One type of thing that we do is fix certain types of errors. This is pretty domain specific, so I won't get into it, but there's a lot of systematically incorrectly published information that we can fix. Another big source of errors is named entity recognition, recognizing all the synonyms and everything of proteins, chemicals. Yes?
I would guess that even without any errors, there'd be a huge combinatorial explosion associated with this assembly process. What do you do with that?
You mean the space of possible assemblies?
Yeah, it would basically be way out of them.
That's a good question. We currently INDRA doesn't really try to enumerate or traverse the space of possible assemblies. I will talk about a probabilistic component that tries to end up with a single high confidence solution, but we haven't yet actively tried to traverse the space of possible assemblies. But we are actually one of the projects that are already funded by DARPA. That's basically the plan, to try to turn INDRA into a probabilistic assembler where different assembly solutions are sampled from the space of possible solutions in a way that they correspond to the prior probabilities of various pieces of information being correct. So right now we don't yet deal with that problem. Okay, so named entities, there's a story there, but the main point about named entities is it turns out that I have hinted at this a little bit a few slides ago, is that you have people talking about specific genes and proteins that actually have these aggregate family categories as well, and sometimes people talk about those, and sometimes there are further levels of embedding. So for instance, PLCD4 is in the PLCD family of proteins, which is in the PLC family of proteins. Here are specific publications that talk about PLC gamma, PLC beta 2, and phospholipase C, right? And unless you can figure out how these things are related to each other, you cannot really consistently assemble knowledge. So we, along the course of implementing INDRA, we built this FAMPlex ontology that tries to deal with this issue for protein families and complexes. And in addition to the hierarchy of these entities, we also have similar hierarchies or ontologies for types of activity of proteins, types of modifications of proteins, and cellular components like the nucleus is inside the cell, for instance, a very simple one. So that we can, given a big set of INDRA statements from all sorts of sources, we can actually find exact duplicates. For instance, IRS1 is dephosphorylated, was mentioned 28 times in 28 different sources. And then find these refinements, like IRS1 is dephosphorylated is a less specific version of IRS1 is dephosphorylated on tyrosine. And then you can keep going up this tree, or well, not really a tree, but a directed graph. IRS1 phosphorylated on a specific site leads to the dephosphorylation of IRS1 on that specific site, right? So this is what you end up with given these various kinds of ontologies. And the ontologies are actually plug and play, so if you assemble information that you need different ontologies for to find these refinements, you can do that too, and you would end up with a different graph. Now, another important point is linking mechanisms, which is because various sources describe things in a way that there are gaps gaps left in the mechanism, and sometimes you can infer out these gaps and fill them in. Yes?
I'm getting a little lost here.
Yeah.
Originally, it sounded like you were trying to get to a different system of differential equations, and now you seem to be talking about getting to an ontology instead. How do these relate?
So this part is about knowledge assembly. So every sentence like this is a fragment of knowledge that came from some source, a paper or a database. And now I'm just talking about making this knowledge base consistent to then prepare it for modeling. Because if you
well, actually, it's going to give semantics to the model.
Yeah, so it's more about the underlying knowledge that will go into the model, and generating differential equations is the last step, right? Okay, so inferring missing information, and then an important component is something I call the belief engine, which is it's a probabilistic model of the error characteristics of different sources, like different reading systems or human-curated databases, and then looking at the aggregate evidence for a given mechanism from all the different sources and taking random and systematic error components into account, and then exploiting the redundancies among mechanisms to propagate these probabilities and end up with a final belief score that tells you overall how confident you can be that a given piece of mechanism is not incorrect. And then we don't have this sampling framework yet, but right now what we do is we take a cutoff and we say, for some applications we might be okay with a low cutoff and include information that was only 75% probability to be correct. With a high cutoff, we can end up with highly precise information, but potentially lose a lot of things that could otherwise be included. So, for instance, a high belief cutoff, you could imagine it as having to have corroboration from multiple sources, possibly partially from human-curated databases. And then once all this knowledge assembly done, you end up with a bunch of these INDRA statements, and that's when you can deal with the specific model assembly. And again, there are many different types of model assemblers. One of them is this that generates these mechanistic differential equation-like models, and there's this issue here about what your assumptions are in assembling a given piece of knowledge into a mechanistic model. The point here is that the user has specific applications in mind, and they can choose a policy for actually implementing this high-level knowledge in a low-level implementation form, and there are various ways you can choose that. Okay, so that's basically how INDRA works, and there are 4 applications that I can talk about, but I don't think there's that much time left, so maybe I can choose one of these and I can answer questions about the others. So one application is actually a human sitting down and writing a paragraph about a molecular mechanism that is automatically turned into a computational model and simulated using INDRA. And the user looks at the output and can rewrite the sentences to test different hypotheses if their initial hypothesis didn't work. There was a news and views written about this when we published this, and it came with this very cute figure about 2 people talking and comparing their hypothesis using this approach. Another is a much larger scale application where we try to automatically explain a high throughput data set, observations in a high throughput data set using reading about 100,000 publications and assembling the information. We are also building a dialogue system that makes use of this natural language modeling technology and enhances it with various kinds of information retrieval. And there's this RAS machine which goes out and reads the literature every day about the specific protein and its neighborhood and figures out how the new information relates to its existing model and updates and publishes the model daily. Is it okay if I talk for a couple of minutes about this application? And just in the interest of time, I'll skip some of the other ones. Okay, so in this application, we have a high throughput data set. These are in a cancer cell line, a melanoma cell line actually. These are all drug treatment conditions that people apply to this cell line, and these are readouts. These are the phosphorylation states, activity states, and amounts of different proteins, and then each of these boxes means that something is going up or going down. And then the question is, you look at one of these boxes and you ask, well, preferably an interesting box, and you ask, how did this happen? What could explain this? So what we did was we read 95,000 papers that are relevant for this data set with 3 NLP systems, and extracted this as a set of INDRA statements, and then added knowledge from these pathway databases, also relevant for this. Here's this figure about access to full text, so 68% abstracts, 12% Elsevier, and about 20% free open access from .NET Central that we have access to. And then we ran this assembly pipeline that I talked about and also filtered it down to the specific use case and built one of these rule-based dynamical models. And then we did structural analysis on the dynamical rules that govern that model. Each of these blue boxes is a mechanism rule. It's like a chemical reaction template, kind of. And the green edges are positive influences from one rule to another, and the red edges are negative influences from one rule to another. And then you can find paths in this graph that have an overall consistent mechanism and polarity that connect a source to a readout. And that's how we can explain the effects of drug perturbations on a given readout. And so for the 22 strongest effects in this data set, we were actually able to find explanations for 20 of them using this approach, again in an automated fashion. And to look at the specific one, one of the things we wanted to explain was how does SARP inhibition increase check to phosphorylation at this specific site. You can display it either as a little network here, but you can also assemble it back into English, as I said, so you get this little paragraph that describes the mechanism that INDRA hypothesizes is the most likely explanation. And we actually take data into account to prioritize between possible explanations. So this is like the top hit that comes out. And the cool thing is that you can actually point to any of these specific edges, and you can say, well, what's the evidence for that mechanism being true? And then you can look at all the provenance for that. So for instance, TP53 decreases PLK1. You have this table of PMIDs and specific sentences that tell you that P53 has been shown to suppress the transcription of PLK1, and so on. So you have full introspection into what happened and where the information came from. Okay, so that's one example of how this can be applied to a high throughput data set. And I'll stop there. There are a number of other applications. I just want to show the acknowledgements slide, which is right. It was right there. Right here. So John Bachman is another postdoc who we work together with on a lot of these projects. Peter Sorger is the head of the LSP and PI on many of these grants, and a number of other people from the lab contributed to various pieces of this work. And we work with our amazing NLP collaborators and other people who build various kinds of pathway databases and network tools. And of course, all the funding comes from DARPA. So. Thank you.
Questions?
I'm imagining that what the data you're trying to get here is really trying to look at steady state data, which is really coming from the system that's assumed to be some kind of a steady state, at least suitably averaged. But the models you get are full dynamical models. So you could look at perturbations and things like that. But also in these sorts of chemical reactors, you might look at them, it's known that even with given inputs and outputs in a steady state, there can be multiple steady states, yes, depending on how it got there. So are these impressions correct? And what do you do about the multiple steady states problem? Do you try to look at that? Or?
Yes, I think a couple of points about that. So one is that we don't necessarily always assemble this information into dynamical models, right? So sometimes for some applications we don't need a dynamical model because, let's say, indeed we are just trying to explain steady state information, and then the detailed dynamics aren't necessarily the main factor that we want to say something about. But yes, the main bottleneck with dynamical models is the parameterization, so figuring out what the weight constants should be that govern some of these equations. In terms of initial conditions, it's actually easier because initial conditions typically map onto expression amounts of proteins in a given cell type that you can measure. The rate constants are much more difficult to measure, so what we would then do is try to take some time course data and calibrate the model to fit that data, holding out some test data to make sure it's not overfit, and then use that calibrated model for further predictions. In this use case that I just talked about, we didn't actually need that because this data set was a single time point assumed to be roughly steady state, and so the more interesting question there is what are the mechanistic paths that can lead to this outcome, and the transient temporal behavior is not really a factor that we need to
to get a steady state model, you have to assume a certain concentration stay constant because they're being replenished by being superior to the model. So you also have to make that decision as well.
Yeah, that's true. To some extent, I mean, these are cell line experiments, so they are in isolation, and then there's a drug treatment, and nothing else is, perturbed in the system. So to some extent, it's isolated in that sense. But of course, there are still various kinds of cellular processes going on. Some cells die, and so on. I think that wouldn't necessarily change the set of possible explanations for a given outcome, but it might modulate which one is the actual likely explanation.
You try to take into account pharmacokinetics.
You mean like how drugs penetrate the cells? Well, not in this case, because this is again in isolation in a patient. So
ideal cells.
Yeah, so that's it's possible that it introduces some artifact, but we haven't dealt with that aspect.
So really, it's an intracellular model, and essentially what I think you're trying to do is, if you look at those big, huge, impressive diagrams of, say, the Calvin cycle that shows all the rate compounds, you're trying to automatically assemble these from the literature.
Yes, and then again with an automated framework, try to explain data with it. So that's one of the ways to look at it. Yeah.
Okay. Very interesting. Very, very ambitious.
So Benjamin, so I'm curious, what stage is this project? Because obviously you've done proof of principles, right? But like, if I were to do a GitHub thing, would I be able to run it in real time? Are the databases updated because you have to kind of lots of? So is that how we're thinking about, or we're still in the proof of principle stage?
Good question. I mean, so it is a usable piece of software, and you can install it and start working with it. And we have collaborators who installed it themselves and worked with it on various projects. I think the bigger question is how general it is, for instance. So whether it generalizes well beyond cancer signaling.
Okay.
And there isn't, the main issue there is, for instance, the reading systems, for instance, are biased towards recognizing human genes and proteins rather than, for instance, yeast genes and proteins. And so if somebody was working on a yeast project, they might not see the most optimal setup for their use case. And so it's rather these kinds of issues that make you say that it's in the proof of principle stage. But we're working on generalizing it. We're working on other disease areas like Alzheimer's. And we're working on generalizing INDRA to completely different use cases, like world models, where we build these kinds of things like review and reports and things like that, and build models of flooding and food production and precipitation. And so, trying to generalize it in that sense. But I think, as far as the software goes, we really try very hard to make it tested and documented and reusable. So to try to go beyond, I think that comes partly from Peter Sorger, who is really into trying to make biomedical research reproducible and reusable, and so we actually spend a lot of time engineering the software to be that way.
Any other questions?
What department are you in HNS? Are you in lab?
So this is the Laboratory of Systems Pharmacology. That isn't really part of a department. It's directly under the medical school, but Peter Sorger used to, he's a professor in the Department of Systems Biology, but his lab has sort of moved out of there and is now directly under the medical school in this sort of special setup. It's almost, you can imagine it kind of like a small department, actually.
How old is the Department of Systems Biology then?
SysBio is like 12, 14 years old, maybe, something like that.
Yeah, that's about right. More questions? Let's thank you.